Hey everybody, we are going to continue on today talking about the macromolecules, and we are going to get started with lipids. So lipids are a diverse group of hydrophobic molecules, meaning that they don't like to interact with water. Like carbohydrates, they are made of carbon, hydrogen, and oxygen, but unlike carbohydrates, they do not have a general formula. So carbohydrates, remember, was CH2O is the general formula. You don't have anything that easy to remember with lipids, but what you can do is look at the ratio of carbon to oxygen, and lipids are going to be much higher in carbon than they are in oxygen. And just not a point here, not strictly speaking macromolecules like the other groups that we're going to talk about, but we do kind of cover them. They're not going to be made of repeating units the same way as the other macromolecules. So very generally, the probably most common lipids are going to be fats and oils. Fats are solid at room temperature, so think like butter, and then oils would be liquid at room temperature, so like any type of cooking oil. They are made of two kinds of smaller molecules. So you've got fatty acids that are attached to a glycerol molecule. Fatty acids are going to be a long carbon chain, anywhere with like 12 to 18 carbons. They're going to have a carboxylic acid group on one end and then a methyl group at the other end. So you can see this right here. So you've got your carboxylic acid group right here. You've got your fat group right here. So this is the fatty acid. This is the fat part, that's the acid part, and then it is attached to a glycerol molecule. And down here, you are looking at a triacylglycerol, so you've got three fatty acids attached to one glycerol molecule. So triacylglycerols is, again, when you have three fatty acids joined to one glycerol, and the bond that joins them together is called an ester linkage, and that happens to occur between the carboxyl group of the fatty acid and the hydroxyl group of the alcohol or the glycerol. We also like to talk about fats in terms of whether they're saturated fats or unsaturated fats. Saturated fats do not contain any double bonds, so we say they are saturated with hydrogen. Everywhere where you could possibly put a hydrogen bound to a carbon, you've got one. With unsaturated fats, you are going to have one or more carbon-to-carbon double bonds, which means that you have to remove a few hydrogens to get those double bonds in there. So technically, an unsaturated fat could accept more hydrogens. What's interesting is that the double bonds cause these kinks in the molecule's shape, so it causes it to have like little bends. So here you've got a saturated fat and fatty acid. So example here would be something like in butter. And now when you're looking at the oil, you can see this is unsaturated. It has a double bond here between the carbons, and it causes the molecule to bend. Alright, so why do fats usually contain saturated fatty acids, whereas oils usually contain unsaturated fatty acids? It happens to be that the double bond pushes the molecules apart. It lowers the density, which then lowers the melting point. So at room temperature, oils are going to be liquid. Alright, so fats. They're going to be different based on the fatty acids that are used to create the fat. And when we talk about fats, they are going to be used for energy storage, they're cushions for organs, and they're also used for insulation. And I said energy storage, so just a quick question. When we're talking about how carbohydrates are used for energy versus fats, so which do you think would have more energy, a kilogram of fat versus a kilogram of starch? And the answer is actually fat, because there are more carbon to hydrogen bonds, which provide more energy per mass. Another type of lipid we're going to talk about are phospholipids. These are similar to fats, but they only have two fatty acids, and then that third hydroxyl group of the glycerol is going to be joined to a phosphate-containing molecule. And so it ends up looking like this. So you are going to have the hydrophilic phosphate head, so this part will like to interact with water, and then you're going to have the hydrophobic fatty acid tails. So those are going to be the areas that don't like to interact with water. And so what happens are these molecules are going to self-assemble into micelles or bilayers, which we will spend a lot of time talking about later on in another chapter, but these are really, really important part of cell membranes. Next, we also have steroids. These are lipids with four fused rings, and they will differ in which functional groups are attached to the rings. So it could be cholesterol, or it could be something like different sex hormones. So again, it's going to be different based on the functional groups that are attached. All right, the next macromolecules that we're going to talk about are proteins, and these are the molecular tools of a cell. Okay, we're going to talk about all their uses, and there's tons of them. Proteins are made of carbon, hydrogen, oxygen, but also nitrogen and even sometimes sulfur. And again, there is no general formula, so look for the nitrogen when you're talking about proteins. So, uses of proteins. Really important for structure. Proteins are enzymes which help the cells run. Antibodies, transport, so they help things move through cells. Other types of movement, receptors, hormones, all right, so lots of uses for proteins that we're going to spend a lot of time talking about throughout the year. Now, proteins are polypeptide chains of amino acids that are linked by, this time we call them peptide bonds. Okay, so it's peptide bonds that link different amino acids together that then form proteins. Amino acids all are going to have a central carbon with 4 attachments. So on one end, the carbon will have a carboxylic acid group. On the other end, the carbon will have an amine group. It will have 1 hydrogen, and then R is just a placeholder. There's going to be some other side group attached to that central carbon. Now, for those R groups, you have 20 different options. 9 of them are going to be creating nonpolar amino acids. 6 of them would create polar amino acids, and then you're also going to have 5 electrically charged. So 2 of them are going to be acidic and 3 of them are going to be basic. We're going to take a look at them right here. So these are nonpolar amino acids. So you can see every single thing that you're looking at has a central carbon, a carboxylic acid on one side, an amine group on the other side, a hydrogen, and then in white is where you see the highlighted R group. So these are going to be your nonpolar amino acids. Here you have your polar amino acids, and then down below your acidic and basic amino acids. So if you are looking at a protein that has sulfur, that sulfur is going to be contained within the R group. And then the properties of R groups are going to be really important because they help to determine the properties of the protein. All right, so polypeptide chains are formed by dehydration synthesis, like everything else we've talked about, but it's going to happen between the carboxyl group of 1 amino acid and the amino group of another amino acid. And so it's going to end up producing a backbone of nitrogen, carbon to carbon, and then that's just going to keep repeating itself for every amino acid that you have linked together. Okay, so here we have the first amino acid, okay, so nitrogen, carbon, carbon, a peptide bond, nitrogen, carbon, carbon. So you've got 2 amino acids joined together. You can see you've got the hydroxyl group and the hydrogen off of the amino group. So through dehydration synthesis, this water is going to leave, and now you have a new amino acid linked to your polypeptide. And so again, nitrogen, carbon, carbon, nitrogen, carbon, carbon. You guys see the pattern there. When we talk about proteins, it's also really important to talk about the structure of proteins. And we actually study protein structure at 4 different levels. So the first level is going to be the primary level, and this is just looking at the sequence of amino acids within the polypeptide chain. And there's many sequences that are possible because you've got 20 different amino acids that you could link together in different orders. The secondary structure of a protein is this 3D structure that's formed by hydrogen bonding between parts of the peptide backbone. So there's going to be 2 main secondary structures that we look at. The first is an alpha helix right here, and then the second is called a pleated sheet, and the example is right there. Next, we've got the tertiary level, and this is going to be bonding that happens between the R groups. So examples for this could be hydrophobic bond. When you've got hydrophobic areas that don't like to mix with water, it'll kind of bend in on itself. You've got ionic bonding, and you also have disulfide bridges that will bond together, and that is right here. So essentially that disulfide bridge is 2 sulfurs that are going to bind together. And then finally we've got the quaternary level, and this is when 2 or more polypeptides unite to form a functional protein. So again, you've got several polypeptides coming together, and a really good example is hemoglobin. Now when we talk about whether or not protein structure is important, a really good example is when you look at blood cells. So in this picture you've got normal red blood cells, And what you're looking at is the primary structure of normal hemoglobin. Now over here, you are looking at sickled red blood cells, and you can tell that the primary structure is different. So you have a change in this number 6 amino acid. And so that one change of one amino acid completely changes the shape of the red blood cells, and it completely changes how they function. So this is just a breakdown of how that primary structure affects the shape of a subunit. And then when you look at quaternary structure, when the different polypeptides come together, they're not going to fit together the same way because of that just one change in one amino acid. We can also denature proteins, and denaturing is essentially any event that's going to cause a protein to lose structure and function. That could happen because of a shift in pH. It could be due to high salt concentrations. It can also happen because of an increase in heat. And so when you denature a protein, it's essentially changing its shape. It almost looks like, you can see, it almost looks like it's unfolding. Certain proteins, once they're denatured, stay denatured. Other proteins, though, will renature, and so they become functional again. Another class of proteins are called chaperone proteins. These are large complexes, and they help other proteins to fold into the correct shape. These are used when cells are stressed, and it helps to keep proteins intact and functioning normally. And so here you can just kind of see it's a large structure. The polypeptide goes inside of the chaperone protein, gets folded, and then it will exit. All right, the final group that we're going to talk about are nucleic acids. These are informational polymers. They're made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So if you're ever trying to figure out what something is, look for phosphorus, and if it has it, you're looking at nucleic acid. Again, no general formula, but two really good examples, DNA and RNA. All right, so nucleic acids, whoops, sorry, too far. Nucleic acids are polymers of nucleotides, and they are made up of 3 parts. They're going to have a nitrogenous base, a pentose sugar, and a phosphate group. Okay, so these nitrogenous bases are going to be rings of carbon and nitrogen. The nitrogen atoms are going to tend to take up hydrogen ions, which makes them a base. And you've got 2 types. You've got pyrimidines, which are a single ring, and then purines, which are double rings. Then your pentose sugar, pentose meaning 5, so it's a 5-carbon sugar. In RNA, that sugar is ribose. In DNA, that sugar is deoxyribose. And these are very, very similar. They are different based on the location of a hydroxyl group on the 2nd carbon. When you're looking at something called a nucleoside, you're just looking at the base and the sugar. When you're looking at something at nucleotide, which is what we're going to spend most of our time looking at, you're looking at a trio of the base, the sugar, and the phosphate group. The PI means inorganic phosphate. So DNA, deoxyribonucleic acid, makes up genes. This is our genetic information that gets passed from one generation to the next. RNA, or ribonucleic acid, is really important for structure and protein synthesis. It does contain genetic information for a few viruses, and it's really important control mechanisms for some genes. We will talk a lot about DNA and RNA in future units, so you guys will get all kinds of information on DNA and RNA to come. But for this unit, for this chapter, you guys should understand the role of hydrolysis and dehydration synthesis. And for each macromolecule, you should know the elements that it contains, its monomers, and then structures, functions, and real-world examples. Okay, so your lecture question for today: when looking at the molecular or structural formula of a macromolecule, how could you tell them apart from each other? So what would you look for in order to see if something was a carbohydrate, a protein, a nucleic acid, or a lipid?